Okay, so hello everyone. Welcome to this time's Immuzoom seminar. So it's really my great pleasure to welcome today's speaker, Dr. Brian Glubransen from Michigan State University. Brian currently is Associate Professor and MSU Foundation Professor in Neuroscience Program, Department of Physiology, Michigan State University. So actually his lab is particularly interested in neuroinflammation in gastrointestinal tract. And then a little bit of brief background about Brian. So Brian completed his undergraduate study in University of Wyoming, and then follow up with his PhD study in University of Colorado, Denver School of Medicine, in Department of Cell and Development Biology. So in 2007, after he completed his PhD study in neuroscience, he decided to pursue his postdoc training in Hotchkiss Brain Institute, Department of Physiology and Pharmacology in Calgary in Canada, particular study in enteric neuron transmissions. So from 2012, after Brian completed his postdoc training, he actually started his independent research program as assistant professor in neuroscience program, Department of Physiology in Michigan State University. And then particular focus in interactions between the neurons and the glial cells in the enteric nervous system. Now, from 2017, he being promoted to associate professor still in the Department of Physiology, Michigan State University. So the goal of Brian's research program is try to understand how inflammation in the nervous system, particularly in neural inflammation, leads to long-term changes in neural circuitry. Within this context, they focus on regulation of neural networks by glial cells, with particular interest in clarifying the role of glial cells in nervous system physiology, and then elucidating how glial cells change during neuroinflammation contribute to the different diseases. And they are addressing this issue by exploring how neuroglial interactions in the internal nervous system regulate intestinal physiology and pathophysiology. Their experiments are designed to let them to understand the fundamental rules that govern cell-cell interactions in the internal nervous system, and then try to illustrate the molecular machinery that contributes to change the enteric network output. It also likely these basic rules in the neuroglial interactions they uncover will actually really broaden the understanding of how neural networks are regulated by glial cells, which I think a lot of us underestimate in other branches of the nervous system, and benefit the development of treatment of non-intestinal nervous system disorders. So with all these particular contributions in glial biology, Brian really got a lot of highlights and then awards, including the David Proud Research Awards, the New Investigator Awards from America's Physiology Society, and then the Beverly Patterson Bishop Awards for Excellence in Neuroscience from 2016. I think I'm particularly impressed when I read through Brian's CV. He really have dedicated in training different trainees, including postdoc, PhD student, and then undergraduate students, and even high school students, to really actually inspire them for their interest in science. And then there's really a lot of achievement from these students. Actually, you can see they are all very active in the field. So I think without further ado, we are really excited looking forward to your talk, Brian. So please carry away.
All right. Well, thank you so much for that great introduction, and thanks for the opportunity to tell you guys about my work. I'm excited to present to this group, and so I'll preface this a little bit by saying, so my group is a neuroscience and gut physiology group, and we're not necessarily immunologists, but since the gut is a huge immune organ, we're dealing with immunology all the time. So I want to tell you a little bit about our work on enteric glia, and these cells in particular have come out as important neuroimmune modulators of the gut. So what we're really interested in is, let me find the, oh, can you all see my pointer okay? Is that okay? Okay, good. So what we're interested in is this group of disorders called disorders of the gut-brain interaction. And these things used to be called functional gastrointestinal disorders. And probably the one that people are most familiar with is this disorder called irritable bowel syndrome. But this includes a wide range of other disorders like chronic intestinal pseudoobstruction, slow transit constipation, and even inflammatory bowel disease in remission has some features of these disorders of gut-brain interaction. And so these are among the most common chronic medical conditions encountered in practice, and they're among the least understood. And we know that it involves altered communication between the gut and the brain, and it involves a host of other factors and susceptibility factors. And what's been really interesting recently is now we've been starting to understand that these disorders of gut-brain interaction are actually features of some of these other diseases that we don't necessarily automatically associate with the gut. And things like diabetes, Parkinson's, obesity, and Alzheimer's actually have pretty debilitating GI symptoms, and we don't really know why that is. So like I was saying, some of these disorders of gut-brain interaction are the most common common disorders encountered in clinical practice. And this is just a summary of incidence of some of these disorders of gut-brain interaction. And so the inner ring shows the incidence of irritable bowel syndrome, so the most common disorder of gut-brain interaction. And the outer ring here shows any functional gastrointestinal disorder or any disorder of gut-brain interaction. So you can see where we are here in the United States, we're really not in a good place with either of these. And so the green here on the inner circle indicates that we're greater than 5% in our population has IBS, and that's pretty consistent with worldwide averages. And the current estimate is that at somewhere around 10% of humans have irritable bowel syndrome. But if you go to this outer ring, we're here in red. So that means that greater than 40% of our population has one of these disorders of gut-brain interaction or one of these functional bowel disorders. And so that's getting close to half of all people basically have one of these disorders. So they're very, very common. And in some people, they're very debilitating. So what causes this? Well, they're caused by a lot of risk factors, and it's a complex interaction between the brain and the gut, diet, microbiome, exposure to many different things. And so we're interested in understanding what causes two of these key symptoms of these disorders of gut-brain interaction. And so these are visceral hypersensitivity, and this is what causes us to have gut pain or abdominal pain, and then altered gastrointestinal motility. So you either go too much or too little. So you either have constipation or diarrhea. So we're understanding the factors that drive these two key factors in these disorders of gut-brain interaction. So the three clearest risk factors for developing one of these things include being female, having inflammation at some point in your life, so experiencing acute gastroenteritis, and experiencing some kind of early life adversity. And so these are gastrointestinal infection here impacting the gut, and these stress and psychological comorbidities affecting the brain and disrupting the gut-brain axis. And so we're interested how inflammation in the gut or psychological stress interact together with female sex to drive some of these disorders. And I'll tell you a little bit about both aspects of work. I'm not going to tell you as much about the early life adversity story. That's a different story than today, but I'll focus mostly on inflammation and how inflammation is driving both altered gut motility and visceral hypersensitivity. So to understand this, we have to understand how the gut works and what drives gut motility and what also is controlling visceral sensitivity. So the gut is actually a really fascinating organ because it has its own brain. And the gut's able to function and perform most of its normal things without any communication with the brain at all. And so you can see examples of this here in these two videos. So this video on the left is showing an isolated piece of guinea pig colon. And if you put a piece of guinea pig poop in there, The colon is smart. It knows which end is front, which end is back, and it can take this piece of fecal pellet and it can coordinate the movement of that fecal pellet down its length. And this is a process called peristalsis. So in this video on the right, this is an isolated piece of guinea pig ileum. And here we've inserted cannulas in the oral and aboral ends of this thing and then pressurized it. And so when you pressurize the ileum, that initiates a different kind of pattern of motor function. And so this is this mixing and churning type activity that's meant to break down food and aid in digestion. But both of these types of motor patterns require no connection with the brain. And that's because the gut has its own brain called the enteric nervous system. And so this is the enteric nervous system is the largest branch of the autonomic nervous system. And this lines within the walls of the gut itself. So within the walls of the GI tract, you have somewhere on the order of several 100 million neurons, and these are surrounded by glial cells. And so these lie in two different neural plexuses. And so the first one of these lies, so this is a cartoon showing a section of gut. And so this is the mucosa that's toward the lumen of the gut. And so deep to the mucosa, you have this plexus called the submucosal plexus. And the neural network there mainly coordinates secretomotor function and secretion and absorption activities across the gut epithelium. So then deeper to that, you have layers of circular smooth muscle and longitudinal smooth muscle. And then sandwiched between these two layers, you have a larger neural plexus called the myenteric plexus. And so the neurons in that plexus innervate circular and longitudinal smooth muscle coats, and they coordinate these activities like shown here in peristalsis. And so this is an example of what one of these ganglia in this network look like. And each one of these green things flashing here is a myenteric neuron. And so this is a mouse expressing calcium sensors in these enteric neurons and glia. The glia here express a TD tomato tag, so they show up as magenta in these images. And so you can see these, in this one ganglion here, there are probably 50 to 100 neurons surrounded by an equal or greater number of glia. And it's these intrinsic networks in the enteric nervous system that are able to coordinate these types of motor behavior. So in its simplest way, we can illustrate one of these simple reflex arcs controlling motility in this diagram here. And so this would be a reflex arc controlling this motor pattern underlying peristalsis. So the enteric nervous system has its own class of sensory neurons, and these are called intrinsic primary afferent neurons, or IPANs. And these neurons send projections up to the gut epithelium where they sense mechanical, chemical, and thermal cues from the gut epithelium. And they're modulated by this special type of chemosensory cells called enterochromaffin cells. So when these sensory neurons are activated, they synapse onto interneurons that project upstream up the gut and interneurons that project downstream down the gut in the anal direction. And these interneurons that project up the gut excite excitatory motor neurons that synapse onto the smooth muscle cells and excite it above this spot. And the interneurons that project down the gut synapse onto inhibitory motor neurons that inhibit the smooth muscle and cause a relaxation below that spot. And so this type of reflex arc is thought to be reiterated over and over down the gut. And so this is what drives a contraction above that spot and a relaxation below that spot that's stimulated to drive a pressure gradient and drive things down the gut lumen. So sensitivity or our sensations of what's happening in the gut too are also mainly driven by nerve fibers that innervate the gut, but these aren't enteric nerve fibers. These are sensory nerve fibers that come from neurons that live in the dorsal root ganglia. And so this is an example of what some of these nerve fibers look like. And this is a mouse that expresses TD tomato under the control of TRPV1. And so these are nociceptive sensory neurons that innervate the gut from the dorsal root ganglia. And these things sense mechanical, chemical, and thermal cues. And in certain circumstances, when these become sensitized, they can convey sensations related to nociception and pain, where we might have abdominal pain or cramping. So the real problem is that when this works normally, it's great. And we all have no idea that this is going on most of the time, even though these processes are going on without us even knowing it, right? But after we have inflammation, this process of motility can be disrupted. And so this is what happens actually in experimental animals. If you give a mouse acute gastrointestinal inflammation, there are changes in the contractions and the relaxations. And so this is some data measuring neuromuscular contractions from the mouse colon. And after this mouse has experienced DNBS-colitis, which is an acute chemical model of colitis in mice, you can see that the contractions here are enhanced and the relaxations are decreased. And if you were to measure propulsive motor activity in a preparation like this with the pellet moving down the gut, normally it would be very linear and things progress down the gut pretty smoothly. But after experiencing inflammation, sites that are ulcerated, these pellets can become delayed. And so you see these spots where there's a delay in transit. Or if it's a severe spot of inflammation, sometimes these pellets can't move past that spot and there can be an impaction. And so these can cause these long-lasting changes in motility that persist well after the initial inflammation is even resolved. So the other feature of inflammation is that this also changes the sensitivity of these sensory neurons that innervate the gut from the dorsal root ganglia. And so this is data from the same type of mouse model. And so this is DNBS-colitis in mice. And then these are measurements of nociceptive nerve activity to different distension pressures of the colorectum in mice. So normally there's graded potentials as you increase the distension of the colorectum, and that these are enhanced after these mice have experienced DNBS-colitis. So there's a sensitization of the sensory neurons. And so these mice feel more pain at lower distension pressures. And this is what's thought to underlie both dysmotility and pain in similar processes in people. So the mechanisms underlying these disruptions are thought to be due to changes in the wiring of the enteric nervous system and changes in the sensitivity of these nociceptive nerve fibers. So the known changes that happen in the enteric nervous system after experiencing inflammation include features that include neuron death. So a portion of these neurons, including these nitrogen neurons, die. The remaining neurons also have changed synaptic properties. And so one of the main features is that there's synaptic facilitation. So these neurons will release more neurotransmitters to a lower pulse than they normally would. So with the neuron death and synaptic facilitation, that causes a massive rewiring of the network and can disrupt the control of gut motility. Now, these afferent neurons also display long-lasting changes in their sensitivity that persist well after the inflammation is resolved. And like in those recordings that I showed you, mice that have experienced inflammation still exhibit higher sensitivity to low distension pressures, and this can cause visceral pain. So we were studying the mechanisms that control this feature of neuron death during colitis in mice, and this causes a loss of these nitrogen neurons. And so these are inhibitory motor neurons that project to the smooth muscle. And when you lose these neurons, you lose some of the inhibition in the plexus, and you also lose the inhibition of smooth muscle. And so there's an overall hyperexcitability of gut circuits when you lose these nitrogen neurons. And you see the same things in other models of colitis. And this is some data from a guinea pig model. And you see this long-lasting decrease in neuron numbers after DNBS-colitis in guinea pigs. And so this persists as long as people have measured it anyway. In this study, it was up to 56 days. We've taken this up to about 4 weeks too, and we don't see recovery. And this is consistent with several different types of colitis in mice, including IL-10 knockouts, the oxazolone model, and the DSS-colitis, where we see this about a 20 to 30% loss of neurons, and it's this nitrogen neurons that are most susceptible. So when we studied the mechanisms that were driving the death of these nitrogen neurons, we actually found that it wasn't a neuron autonomous process. We found that this was actually driven by high levels of ATP that activated P2X7 receptors that are expressed by some of these neurons. And so this is some labeling from humans showing P2X7 receptor immunoreactive neurons in myenteric ganglia. And when these neurons were activated by high levels of ATP and triggered these P2X7 channels, receptors to open, this triggered downstream pannexin channel opening and caused ATP release. So what the interesting factor in this was that the ATP that was released from these neurons actually activated all the surrounding glial cells. And these glial cells exhibited these strong calcium responses when neurons were activated with P2X7 agonists. So the mechanisms that drove neuron death ended up requiring signaling through the glial cells, where ATP released through these neuronal pannexin channels was broken down to ADP that triggered calcium responses in glia, downstream opening of these channels called connexin 43, and ATP release that fed back on neurons to kill them. So we thought, gee, these glial cells are really important in driving this inflammatory component in the enteric nervous system and neuron death. What else could they be doing? And they seem like a pretty important cell type. So it turns out that the glia in the gut are actually a special type of peripheral glia. And so they display some similarities to other types of neuroglia, such as astrocytes in the brain. And you can see the physical similarities here by this drawing from Santiago Ramon y Cajal of a cerebellar neuron being embraced by an astrocyte. And we see the same type of relationship between some glia and neurons in the gut, where this is a submucosal glial cell embracing one of these submucosal neurons in the mouse. So glia are labeled here with GFAP surrounding all these neurons in a mouse myenteric ganglion. And then here they're labeled with the TD tomato reporter. And you can see many different glial cells and inside the plexus and outside the plexus. So these glial cells have some similarities to astrocytes, but they're also unique. We know that they come from the neural crest and that they express some markers of astrocytes like GFAP, vimentin, S100 beta. They also express some markers that are typically thought of as markers of oligodendrocytes like SOX10, and they display some similarities with other peripheral Schwann cells. But they're unique both in their physiology and their molecular makeup, so they truly are a unique type of glial cell. So there are several types of these glia in the gut so far that have been grouped in different subtypes. And so far, these have been broadly classed into 4 main groups. So the first one of these groups is called mucosal glia. And this is what these cells look like in a reporter mouse. But these cells live out at the level of the epithelium of the gut. And they're thought to support epithelial health, regulate mucosal immune responses, and probably interact with some of the nerve processes that project out to the gut epithelium. So as we move deeper through the gut wall, the next population of glia that you come to are these glia in the submucosal plexus that we refer to as submucosal glia. And so these glia sit with neurons in the submucosal plexus, and they're thought to regulate submucosal neuron activity and health. And we know that these glia actually regulate secretomotor function through signaling with the submucosal neurons. So the next class is the largest class of glia in the gut. So these are the glial cells that are in the myenteric plexus. And so these myenteric glia, there are many of these, as you can see in this image here, surrounding the myenteric neurons in ganglia. And they're also in these thick fiber bundles that connect myenteric ganglia. And so we know the most about these type of glial cells because these have been the most studied. And so we know that these support myenteric neurons, they regulate myenteric neuron activity, and they contribute to regulating gut motor functions through interactions with neurons. And what I'll tell you about a little bit more today is that we know now that they modulate immune responses at the level of the myenteric plexus. And we think that they have pretty diverse signaling functions. So the last type are these cells that we refer to as intramuscular glia. And these are glia that sit with nerve fibers that run in the circular and smooth muscle coats of the gut. And we really have no idea what these do. Nobody has done a specific function of these glial cells. We think these probably function like Schwann cells with nerve fibers, but we really don't know. It's kind of an open question at this point. So the myenteric glia really caught my attention right away because of some older data from this guy, Giorgio Gabella. And Gabella had some data from the early 1970s. And so this was electron microscopy data showing that nerve endings contacted glia in the myenteric plexus of the gut. And that these nerve to glia synapses were so common that every glial cell in the gut had at least one of these types of synapses, at least in the myenteric plexus. And so these were presynaptic specializations with vesicles in these nerve endings with no obvious specialization in the glial cell. And Gabella didn't really know what to make of these at the time. And there wasn't a good way to test what the physiological significance of these nerve to glial synapses was until we had calcium imaging as a way to study glial activity. So glia are electrically inexcitable cells. So if you study these with electrophysiology, they're pretty boring. But once we had calcium imaging, we were able to study activity because glial cells fire calcium spikes, and calcium signaling is one of the main forms that glial cells use to convey activity in the cell. So when I was a postdoc in Keith Sharkey's lab, we tested this idea and we stimulated some of these nerve bundles innervating myenteric ganglia in the guinea pig. And what we found was that most glia actually responded when we stimulated nerve fibers. And we were able to block this with typical neurotoxins like tetrodotoxin. So more recently, we've been studying this using a little bit more advanced methods. And so we've developed some transgenic mice that express GCAMPs in neurons and glia. And this particular model shown here expresses TD tomato in glia and GCAMP in the neurons and glia. And in this experiment, we used electrical field stimulation to drive activity in the neurons. And so what you'll see in this video is the neurons light up first, followed by calcium responses in all these glial cells that surround the neurons. So we've been interested in what this means and what the significance of neuron to glia signaling is in the gut. And so when we've tested this, we've used several different methods to test this. And so these methods have really been relying on understanding the signaling cascades that the glia use. And so one of the first things that we found was that many of the signaling cascades that trigger calcium responses in these glia rely on GPCR signaling, and that they're very responsive to some compounds like purines and ADP. And so this is an example of calcium responses triggered by ADP in myenteric glia. So downstream of these calcium responses in the glial cells, we found that that triggers opening of these membrane channels called connexin 43. And these glial cells express these connexin 43 hemichannels on their surface. And when they're activated, they can release neuroactive or signaling molecules like ATP. They're used as a signal between the glial cells, each other, and between glial cells and neurons. So the first thing that we did then was we tested if we blocked this important signaling function of glia, what was the effect on gut motility? So we developed mice where we ablated these connexin 43 channels from glia. Shown here, we were able to show that there was nice labeling of connexin 43 in control animals, and this was lost in the transgenic mice when we ablated the connexin 43 channels in glia. And then we studied gut transit and neuromuscular function in these mice. And what we found was that the mice that lacked the glial connexin 43 channels were constipated. So they had longer colonic transit. And then when we did more detailed studies of neuromuscular contractions, we found that the mice lacking glial connexin 43 channels had impaired contractions and impaired relaxations. So the glial signaling was doing something to enhance neuromuscular function in the gut. So then we developed a second model where we could, instead of blocking the function of glial cells, we could stimulate the activity of glial cells. And so we took advantage of knowing that these glial calcium responses were triggered by GPCR signaling. And so we expressed a GQ dread receptor called HM3DQ in enteric glia. And so these dread receptors are designer receptors that are activated by designer drugs. And so the drug that they're receptive to is this biologically inert compound called clozapine N-oxide. And this mimics a physiological receptor pathway expressed by these glial cells because the glial cells actually express M3 muscarinic receptors, and they respond to muscarine. And this is blocked by an M3 receptor antagonist, this J104129. And that's important because this HM3DQ dread receptor is a modified muscarinic receptor. So when we expressed these receptors in glia, we were able to show that puffing CNO onto the ganglia drove these large calcium responses in glia, and that these were followed by responses in the surrounding neurons. So then when we did the same types of experiments and we measured neuromuscular contractions, what we found in wild type litter mates was that clozapine oxide really had no effect. So it was not having some kind of off-target effect. If you stimulate the neurons with electrical field stimulation, that drives a contraction. And if you stimulate the muscle directly with the muscarinic agonist botanicol, that can drive an equal contraction. So you're bypassing the nervous system and just stimulating the muscle. So in the transgenic animals, what we found was that electrical field stimulation drove a contraction, stimulating the muscle with botanicol drove a contraction. But then what was really surprising was when we just stimulated glia with CNO, that drove a contraction of equal magnitude as stimulating the muscle or the neurons. And we found that that was all transduced through neural pathways because we could block that with tetrodotoxin. So the glial cells were activating neurons that then were able to activate the muscle. So then when we studied this in vivo, we found that stimulating the glia decreased colonic transit time. So they pooped faster and they pooped more pellets per hour. And when we measured gut motility in vitro and we studied propagation of these colonic migrating motor complexes, we found that stimulating glia made these colonic motor complexes propagate down the gut faster than they would in control animals. So with these two models, we were able to show then that either blocking the ability of glia to signal to other cells impaired gut motility, and stimulating the glial signaling pathway enhanced motility. So lately we asked, how does that work and why are glia having this excitatory effect over these motor circuits? And so this is recent work that we just published last year in PNAS that was work from a PhD student in my lab, Mustafa. And his project focused on this idea that there may be specific subpopulations of these glia devoted to these ascending and descending arms of the motor pathway. And so he tackled this by stimulating either in the ascending direction or in the descending direction with electrodes, and then recorded the calcium responses of the neurons and glia that were activated in each circumstance. And what he found was that normally there were more neurons activated in the ascending pathway than the descending pathway. And this is consistent with what's known in the field, and that there were many neurons that were activated in both directions. And these are probably these multipolar sensory neurons that are activated by either ascending or descending stimuli. So the glia were also mainly activated by both directions. About half of them were activated in both. There were about a quarter of cells that didn't respond in either direction, but then there were these two populations that were equal and non-overlapping subpopulations that were only activated by this ascending pathway or only activated by the descending pathway. And we think that these are glia that were specifically associated with these interneurons and interneuron synapse pathways that project either up or down the gut. So this is basically our evolving model for how we understand the communication between the glia and the neurons in the myenteric plexus that control motility. And there are these specific subpopulations of glia that are either devoted to the descending pathway or the ascending pathway, and then a large population that's both sensing stimuli in both directions that's activated by these sensory neurons. And some of these neurotransmitters activate the glia, so things like ATP and acetylcholine. And these transmitters, and I'm not showing that data to you today just for the sake of time, seem to act through glia. Acetylcholine seems to act as a cross-inhibitory signaling molecule through glia, where it seems to restrain the surrounding pathways, where ATP seems to potentiate these ascending arms of the reflex pathway. So we're starting to get a handle on what the glia do in physiology, but we're still really interested in what they might do in these situations that give rise to gut pain and abnormal motility. And so, like I was telling you earlier, the glia were involved in this inflammation and neuroinflammation that was driven by DNBS-colitis in mice. And in a separate study, we actually showed that if you blocked glial signaling, we were able to block neuroinflammation in the enteric nervous system. So we thought, well, glia are probably this main driving force of the inflammatory environment surrounding the neurons. And so we wanted to understand what the pathways were that were activated in glia then that could contribute to this inflammatory environment. So we isolated RNA from the glia in control and DNBS animals, and then did RNA sequencing on these. And so several of the things that popped out as top pathways were sensory perception of pain, cytokine activity, antigen processing and presentation, and immune responses. And so these were all things that suggested to us then that glia were having a role then in modulating the nociceptors and contributing to visceral pain, and that glial cells could be contributing to modulating the local immune environment of the gut. So this then raised the question of, so how are glia influencing the neurons? Is this either direct or indirect? Because they both have this seemingly gene profiles that suggest that they influence visceral pain, but they also influence the immune responses. So I'll tell you a little bit about both aspects of this and evidence for both direct and indirect pathways. So these nociceptive nerve fibers that come from the dorsal root ganglia that sense visceral pain innervate the myenteric plexus, and about a quarter of these nerve fibers that come from the dorsal root ganglia project into the myenteric plexus. So when they get into the myenteric plexus, they actually are surrounded by enteric glial cells. And this is a really tight relationship where these nerve fibers are very, very close to surrounding these glia. And so we know that the glia and nociceptors talk to each other. And we know that some of the cytokines that are active during DNBS-colitis can activate both cell types. So looking into the glial sequencing data a little bit deeper, we found that some of the key cytokines that were active during acute colitis were things like IL-1 beta and IL-6. And these were also cytokines that were increased at the whole colon tissue level in both male and females during the time course of DNBS-colitis. So Wilmery, who's a PhD student in my lab, has been asking then how activating glial cells with these cytokines under the conditions of inflammation might affect their signaling with nociceptors. So if you guys remember, one of the targets that glial cells use to communicate with each other is this channel called connexin 43. And we can use this simple assay of ethidium dye flux across the membrane because these connexin channels are permeable to ethidium bromide. And so this is a simple assay to assess connexin 43 permeability in cells. And what she knows is that, so if you stimulate glia with ADP, that normally increases the permeability of cells because ADP activates these calcium responses that lead to connexin 43 opening. This is potentiated in the presence of IL-1 beta, and we can show that it's blocked with a mimetic peptide that blocks these connexin 43 channels. It's not blocked by blocking neuronal pannexin channels. And it's also blocked by this pore blocker gadolinium. And this whole process is completely obliterated in these transgenic animals that don't express connexin channels. So these pro-inflammatory cytokines like IL-1 beta can potentiate the ability of glial cells to communicate with each other by effects on connexin 43. So then Wilmery has developed a new model then to study these interactions between glia and nociceptors. And so she's expressed GCAMPs in the nociceptive nerve terminals. And so she's studying calcium responses in these tiny nerve terminals then that innervate the myenteric plexus. So these are TRPV1 positive neurons, and they respond to capsaicin, which is the active ingredient in hot peppers. So normally if you expose these nerve fibers to capsaicin, that triggers a calcium influx through these channels. In the presence of IL-1 beta, this is not statistically significantly larger than control preparations. And if she activates glia with ADP prior to activating neurons with capsaicin, that has no effect on the sensitivity of these channels in control conditions. But if she does the same experiment in the presence of IL-1 beta, activating glial cells with ADP before activating neurons with capsaicin causes this huge potentiation of the neuronal TRPV1 channels. And she found that this is entirely dependent on glial connexin 43 channels because she can block it with the connexin mimetic peptide 43-gap26. So in the conditions of inflammation, IL-1 beta is potentiating these connexin channels, and it's increasing the ability of glial cells to release something then that potentiates these nerve fibers that transduce pain. So then she studied what the significance of this was in a whole animal model. And so in this experiment, she's giving these animals an enema of IL-1 beta. And these are either wild type mice shown on the top, so wild type female and male mice with a IL-1 beta enema. And what was interesting here was that in male mice, they're not very sensitive to IL-1 beta at all. It doesn't seem like it causes potentiation in a normal male mice. Female mice, it causes potentiation. Their females are more sensitive to IL-1 beta. And we see this visceral hypersensitivity at low distension pressures. So she repeated these experiments in mice that lacked the glial connexin 43 channels and found that she was able to completely obliterate this hypersensitivity to IL-1 beta in females. So this glial connexin 43 signaling pathway is important for sensitizing the nerve fibers by pro-inflammatory cytokines like IL-1 beta. So then what's the mediator that's driving this? Well, she looked back in the RNA-seq data and she found that the gene encoding COX-2 was highly upregulated in glia during inflammation. And we know that one of the products of COX-2 in glia is prostaglandin called PGE2. So Wilmery repeated this experiment now using a mouse that expresses the G camp in the neurons and then expresses the dread receptor in glia. So we've combined these two models together to get a more specific activation of glia and then record activity in the nerve fibers themselves. So now when she does this, she sees the same potentiation when she activates glia with CNO in the presence of IL-1 beta. She's a potentiation of the capsaicin response. And then she's able to block this with an EP4 antagonist called L161928 that blocks the EP4 receptors that are expressed on the nerve fibers for PGE2. So the glial cells are potentiated through this connexin pathway. They release PGE2 that acts on EP4 receptors that can potentiate these nociceptive nerve fibers and contribute to pain. So that's one way that the glial cells can affect the nerve fibers directly, but how about this whole host of genes then that was changed that related to immune responses? Well, that suggested that there's also indirect pathways where the glial cells could influence the nociceptors. And we've been studying that in several different ways. And so I wanted to tell you a little bit about the different types of immune cells that we've been studying interactions with glia. So the first one are the muscularis macrophages. And so this is a special type of macrophage that's similar to microglia in the brain. And these lie around the myenteric plexus in the smooth muscle coats. So what Vlado, who was a recent postdoc in my lab, is now at the New York Institute of Technology. He was studying acute and chronic DSS-colitis in mice using the same connexin 43 knockout model. And what he found was this had no effect on acute DSS-colitis, visceral hypersensitivity in mice caused by DSS, but it was able to block the chronic colitis that was established after multiple cycles of DSS in mice. So when he studied cytokine and chemokine panels in these mice at the whole colon level, the main thing that stuck out to him was this compound called MCSF. And MCSF was increased during acute colitis in the control animals, and that was lost in these animals that where connexin 43 was ablated in the glia, and this was still down in the chronic colitis. And we also saw that MCSF was increased in humans with Crohn's disease in myenteric ganglia, suggesting it might actually have some relevance to humans. So MCSF is an important cytokine for macrophages, and we know that these muscularis macrophages have a tight physical associationship with the glia. So Vlado started studying whether the glial cells could be influencing muscularis macrophage phenotype. And when he studied this using CD68 labeling, he found that during acute colitis, there was not really a significant change in the CD68 labeling, but during chronic colitis, there was an increase in the CD68 labeling of macrophages in control animals, and this was lost in these animals when we ablated connexin 43. So Vlado then isolated cells from the myenteric plexus to see which cells could be making the MCSF, whether it was coming from the neurons or the glia. And what he found was that most of the cells that were actually expressing this membrane-bound portion of MCSF, that's the biologically relevant form, are actually glia. So most of these cells, about 60% of the cells were glia, only about 20% were neurons. So most of the cells expressing this biologically active form of MCSF in the myenteric plexus were glia. So I'll just summarize a lot of Vlado's data here, but what he was able to show was that activity in the glial cells that was mediated through connexin 43 was causing this membrane-bound MCSF to be cleaved. That was causing these macrophages to transition into a pro-inflammatory phenotype, and then that was causing sensitivity of these visceral afferent nerves, contributing to pain. And so this is an indirect mechanism then where glia could influence nociceptor sensitivity through effects on macrophages during chronic colitis. So how about these other types of immune cells then? So another one that's heavily implicated in IBS are the mast cells. And mast cells are one of the most studied cell types in IBS, and they seem like they contribute to sensitizing visceral nerves. So here we used a model actually of early life stress. And so this is an early life stress model called neonatal maternal separation stress. And it's basically like putting mice through daycare where the pups are separated from their moms for 3 hours a day and then return to their moms, and then they're weaned 3 days early. And these mice end up growing up, and they have a phenotype that's actually similar to IBS, where they express visceral hypersensitivity and some gut dysmotility. So when we started looking in these mice at where the mast cells were using a mast cell reporter mouse, we found that there were a few mast cells surrounding the myenteric plexus in normal mice, but there were more of these mast cells, and they were closer to the myenteric plexus in these mice that experienced early life stress. And what was interesting is this actually seemed to have a sex-dependent effect where males seem like they express more of these mast cells than females do. And they're closer to the ganglia in males than they are to females. So we studied what this interaction might be. We started studying what the mast cell supernatants did to the glia. And so these were calcium imaging experiments where we were taking either inactivated mast cell supernatants or activated mast cell supernatants and squirting these onto preparations of glia. And what we found was that these normally the number of cells that were activated by these mast cell mediators was actually lower in these mice that had experienced early life stress. So these mast cell components that they were releasing seemed like they did have some effect on glia where they were triggering calcium responses, but this was actually decreased by these mice that were experiencing early life stress. So one of the mediators that seems like it's important in this mast cell glia interaction is histamine. And the glia actually perform an important role in taking up and degrading histamine in the enteric nervous system. And the number of glial cells and concentration of histamine seems like it's changed following early life stress. And these glial cells seem like they have a higher concentration of histamine. And we also observed some changes in the H1 histamine receptor that's expressed by some of the sensory nerves that project to the gut. It's thought to be an important mechanism of sensitizing the TRPV1 positive neurons. So these types of interactions between the mast cell and glia that are mediated by histamine, we think might impact the sensitivity of these visceral nerve fibers in the context of early life stress. So these have been both innate immune cells, but how about some of these adaptive immune cells that live in the gut, like T and B cells? Well, if you remember, one of the gene profiles that was highly upregulated during inflammation related to antigen processing and presentation. And actually, one of the classic observations in Crohn's disease is that these glia transform into antigen-presenting cells and start expressing MHC class II. And so this has been something that's been around in the literature since the late 1980s. And so we wanted to get at what the significance of this was. And so Aaron Chow, who was a recent graduate from my lab, developed a mouse where we knocked out the glial MHC2 receptors using a SOX10 promoter and a floxed MHC2 gene. And so normally in mice, the MHC2 expression is in some of these muscularis macrophages, And you can induce expression in glia when you expose glia to LPS. And you can see some of the MHC2 labeling here on these glia after exposing them to LPS. In the knockout mice, the macrophages still express MHC2 in both control and LPS conditions, but the glia don't. So they've lost their ability to upregulate MHC2 in this knockout model. So Aaron was using flow cytometry to study what the impact of knocking out glial antigen presentation was. And what he found was that this had a really drastic effect on the activation of CD4 and CD8 T cell populations in the gut. And so normally there was an increase in the proportion of CD69 positive CD4 cells and the proportion of CD69 positive CD8 CD69 positive, CD8 positive cells after exposing mice to LPS. And this was lost in these mice that lacked glial MHC2 antigen presentation. So when he started looking at what some of the profiles of these cells were that were affected, one of the two most that were impacted were these proportions of cells that expressed IL-17 and the cells that expressed FOXP3 that are known as Tregs. So we think that most of this effect was probably mediated on these Tregs and these immune cell populations that are involved in tolerance. So one of the interesting things about the glial antigen presentation was we thought that the glia would be actually picking up pieces of LPS or picking up debris and then presenting it on MHC2 and then presenting that to these lymphocytes. What Aaron actually found was that we couldn't find any evidence of glia autophagy at all, but what he found was that, glial phagocytosis at all, but what he actually found was that glial antigen presentation was actually driven by autophagy, and that he found an increase in some of these markers of autophagy, such as consumption of P62 and an increase in LC3 levels, at the same time as he started seeing this increase in glial MHC2 expression. And he was able to show that these drugs that either activate or inhibit autophagy were able to either activate or suppress the expression of MHC2 in this model. So we think that glial antigen presentation was actually driven by autophagy and not phagocytosis of material. And then this was impacting immune tolerance populations in the gut. So the last little bit here that I just want to touch on in the remaining minutes is a news story that we just published in Mucosal Immunology. And this is interactions between glia and neutrophils. So we actually kind of stumbled upon this by studying how glia were regulating purines during inflammation. Now, if you remember, purines are one of these compounds that's generated at high levels during acute inflammation. And this was one of the signaling molecules that was involved in driving neuron death in the gut through the P2X7 pathway. But as the inflammation insult wanes, you get a decrease in ATP levels. And at the same time, there's an increase in the adenosine concentration as ATP is broken down into its metabolites. So this is thought to drive these different phases of inflammation where early on you have this pro-inflammatory phase that's driven by P2Y and P2X mediated effects through ATP. Then later on, you have this anti-inflammatory pathway that's driven by adenosine. So glia express this type of adenosine receptors called A2B receptors. And it's been proposed in the literature that these A2B receptors are anti-inflammatory. And so this was the model that we had proposed going into this, where we thought that the purinergic P2Y1 pathway was going to be pro-inflammatory, whereas the A2B pathway would be anti-inflammatory, and these would oppose each other's actions in the glia. So we created a glial A2B receptor knockout mouse model, and it worked well at ablating glial A2B receptors, as shown here with this immunocytochemistry. And then we put these mice through DNBS-colitis to study how knocking out glial adenosine receptors affected the inflammatory insult itself. So what we found was that it seemed like knocking out glial adenosine receptors, A2B receptors, didn't really have much effect on the gross macroscopic damage and weight loss experienced with DNBS-colitis. These mice seem to exhibit similar patterns of weight loss and weight gain, although they seemed like they gained weight maybe a little bit better than the controls. And they seemed like they had the same type of microscopic and macroscopic damage in the gut wall. So it didn't seem like we were having a major effect on the gross inflammatory drive in these mice. But when we started studying gut function, we found that that was drastically altered in these animals. So one of the features that seems to drive persistent gut function after inflammation is a change in gut barrier function. And a leaky gut can cause passage of bacteria and other luminal substances that can perpetuate inflammation and cause long-lasting changes in gut function. So what Vlado found was that in this in the resolution phase of after DSS colitis, animals that had experienced DS colitis still had a leaky gut. And you could measure this with FITC in the serum in vivo, or you could measure this in oosing chambers in vitro, and you could measure this leaky gut phenotype. And what was really interesting was when we knocked out the glial adenosine 2B receptors, they were protected from this leaky gut phenotype. So this is actually a beneficial thing of losing the glial adenosine receptors instead of a detrimental thing like we thought it might be. So then we thought, well, what's actually changing in the gut epithelium? And so Vlado ran a panel of some of the tight junction proteins and genes encoding tight junction proteins. And he found that some of the largest effects were actually in Claudin 1 and Claudin 8, where it seemed like there was an upregulation of Claudin 1 and a downregulation of Claudin 8 in the control animals during this resolution phase where they had the leaky gut. And this was partially protected in some cases, or completely protected in the case of Claudin 1 by knocking out the glial A2B receptors. This also affected localization of these tight junction proteins, where in the resolution phase after DSS colitis, there were these clumps of occludin expression in the mite and control animals that had experienced DSS, where this was more of a uniform distribution in the mice that lacked glial A2B receptors. So the glial adenosine signaling was having some effect on barrier function that was mediated by these changes in tight junction protein expression and localization. So to get at the mediators and mechanisms that were linking these two, Vlado studied some of the cytokines and chemokines that were altered in these mice. And one of the ones that really struck him right away was CXCL1. And so this was increased during the peak of colitis, and it remained increased during the resolution phase of colitis. Now, the other interesting one was IL-17. This was not increased during the peak of colitis, but it was increased during the resolution phase of colitis. And both of these were completely normalized by knocking out glial A2B receptors. So when Vlado looked at the ability of glia to actually make these things, what he actually found was that glia seem like they have a good ability to make CXCL1. So this is glial translating RNA from mice that had experienced DNBS-colitis, where they show an increase in CXCL1. They also show this ability in isolated cells. So these are primary enteric glia in culture exposed to IL-1 beta. And CXCL1 is a huge increase, where it's about 1,000-fold increase as opposed to some of these other things like CCL11, which is only maybe 2. So Vlado started studying then the immune profiles that might be affected by these cytokines that were being made by glia. And what he found was that overall, the number of CD45 positive cells was similar in control and knockout animals, and that there were similar proportions of Ly6G positive cells, CD3 cells, and 17 positive cells. But when he looked a little bit deeper into these IL-17 positive cells, he found that the subset of cells that were IL-17 Ly6G double positive cells was lower in the animals that lacked the glial A2B receptors. So this IL-17 Ly6G double positive population is typically thought of as neutrophils. And so Vlado tried to do an experiment where he then used antibodies to deplete neutrophils during the resolution phase of colitis to see if that normalized permeability. And now what he found in this experiment, unfortunately, there was very high variability in this experiment, but it seems like there were specific groups that were up and down in the isotype control. So it seems like the isotype control alone was having a pretty drastic effect on permeability. So this was a confounding factor in this experiment. But it seems like the mice that were treated with this neutrophil depleting antibody and the A2B mice were actually normalized to the control levels. And this is consistent with the complex role that some of these cytokines have on glial cells themselves. And so it seems like blocking glial A2B receptors has a pretty massive effect on the expression of these cytokines and chemokines without any other perturbation at all. So just in the presence of an A2B receptor antagonist, it seems like glial cells in culture upregulate CXCL1, and then this is similar in the presence of this A2B receptor agonist, NECA. And it seems like in the presence of IL-1 beta can increase the expression of CXCL1 in glia, but this is also increased to a certain extent by just blocking the A2B receptors themselves. So this might be a confounding factor in these experiments where the mice lacking the glial A2B receptors have a dysregulated immune profile to begin with. And we think that these immune factors and these glial A2B receptors seem like they have a very complex role in regulating the cytokine and chemokine responses from glia, where glial A2B receptors seem like they have a role in increasing IL-6 expression, but they decrease CSF3, decrease CXCL1, and decrease CXCL10 expression. I think that this can impact a number of different immune cells, including some of these neutrophils, and then this can cause this change in the tight junction expression and localization that can lead to some of these persistent barrier defects that we see in these animals. So I just want to finish with this summary slide then of how we're starting to understand the interactions between glia and immune cells in the gut, where they can influence a diverse number and mechanisms affecting immune cells. And we think that this can impact things like visceral pain by interacting with the macrophages and mast cells, can impact barrier function by modulating neutrophils, and can impact overall susceptibility to inflammation by influencing some of these immune cell subsets involved in immunotolerance. So it seems like it's much more complex than just a either or situation where the glia can either have these direct effects or indirects on the immune cells. It seems like the glia have a very complex role where they're signaling with neurons and modulating enteric neuron activity and gut motility, and these bidirectional interactions are important in regulating normal functions. They also have bidirectional interactions with immune cells where they're regulating immune cell phenotype and activity. And then these can either have effects on visceral pain through immune cells, or the glia can have bidirectional interactions with the visceral nerve fibers themselves that can influence enteric neuroinflammation and visceral pain. I just want to finish by thanking all the people in my lab that do the work. I have a great team of people. Will Murray, who's a pre-doctoral student, did all the nerve fiber recording work, and Mustafa has done all the work with neuron subgroups. Jack is doing all the work with early life stress. Bia is starting to work on motor patterns. Visha is working on a signaling system through LPA. And I think that's everybody who I've got on the slides. And I just want to thank my funding sources from the National Institutes of Health and Michigan State University. And I thank you all for your attention, and I'd be happy to answer any questions you might have.